The overall goal of this protocol is to differentiate human pluripotent stem cells into fully pigmented, mature melanocytes.
This method can help identify key factors during melanocyte development. Additionally, this protocol will enable us to identify factors that have gone awry during disease development, as well as screen for important drugs and during drug discovery. The main advantage of this protocol is it enables us to produce large numbers of fully pigmented melanocytes, whereas isolating melanocytes from patients usually results in relatively few cells.
After aliquoting and freezing gelatinous protein, thaw and resuspend a 1 milliliter aliquot in 19 milliliters of DMEM F12. Then, introduce 5 milliliters of this mixture onto a single 10 centimeter tissue culture dish. After preparing several such plates, proceed to incubate them for 1 hour at room temperature. Next, begin to prepare plates coated with PO, laminin, and fibronectin by first adding PBS containing 15 micrograms per milliliter PO to a culture dish. Then incubate the plates overnight at 37 degrees Celsius in a humidified incubator. Afterwards, aspirate the solution and wash the dishes 3 times with 1x PBS. Continue coating by adding 10 milliliters of PBS containing 1 microgram per milliliter mouse laminin 1 and 2 micrograms per milliliter fibronectin. Then, incubate the dishes as previously described. To begin, add 0.1% gelatin in PBS to a 10 centimeter dish.